Hello, grade 8 students. Here is the summary for what we covered in week 2, and the topics included are self and non-self, cells, molecules, and organs of the immune system. Let's start with application 1, which is related to lesson self and non-self. Okay, doctors have known for many years about transplanting body parts such as blood vessels, heart, kidney, or liver from a living organism to another or inside the same organism. Some grafts were accepted, others were rejected. In their search for the factors that lead to the rejection of the graft, they conduct the following graft experiments A, B, C, and D, and the results were summarized in the graph below. Pose the problem question. So here, what is the problem? What they are searching for? The problem is what are the factors that lead to the rejection of the graft? That's why they are doing these experiments, and don't forget to put the question mark at the end. Pick out the organs that can be grafted from the text. The organs are blood vessels, heart, kidney, and liver. Part C, referring to your acquired knowledge, determine the type of each graft. So, determine means we have to justify, then to answer. So we have to justify, then to say which type of graft in the figure. Let's start from experiment A. We can say, since in experiment A, the graft is done from one side to another in the same body of a rabbit, then it's an autograft. The same thing in experiment B, since in experiment B, the graft is done between two identical mice, as you see in the figure, so this is an isograft. Since in experiment C, the graft is between two organisms, two rabbits, of the same species, then this is an allograft. And since in experiment D, the graft is done between two organisms of different species, a mouse and a rabbit, so this is a xenograft. Okay? Analyze all conducted experiments. Analyze, we have to mention the variable and the result. We can start from anything, variable and result, or the opposite. Let's start from the experiment, from the variable here. We can say, we will analyze experiments A and B together, and you will do the same thing for C and D. Experiment A. Upon doing a graft from one side to another in the same body of a rabbit, the survival of grafted cells remains constant 100% as time passes from 0 to 2 years. Okay, as you see in the graph here. B. After doing a graft between two identical mice, the survival of grafted cells decreases from 100 to 80% as time passes from 0 to 2 years. E. Derive a conclusion concerning the fate accepted or rejected of each graft. We say, we conclude that grafts A and B were accepted while grafts C and D were rejected. Why? Since in grafts C and D, the percentage of survival of grafted cells decreases to approximately 0 after 2 years. Okay, part F, using all previous documents, draw out the suitable conditions for the success of the graft. The success of a graft depends on the similarity of MHC between the donor and the recipient. Tabulate the obtained results. So we draw a table. This table shows the percentage of survival of grafted cells at year 0, after 1 year, and after 2 years in each of the 4 grafts. So the title is a table showing the variation of the survival of grafted cells as a function of time in years in grafts A, B, C, and D. Activity 2, cells, molecules, and organs of the immune system. The first thing you have to know is the meaning of infection. Infection is the invasion and multiplication of a non-self inside the body. Let's read document 1 to learn more about the cells that fight the non-self. Immune cells are our army cells that defend against all non-self cells using different strategies. They are the white blood cells, which are also called leukocytes. Some of them, like macrophages with horseshoe nucleus just eat the bacteria cell and break it down. Others, like granulocytes with multi-lobed nucleus, have similar function as well. All of the mentioned cells need to cross the capillary wall to reach the infected area in order to eat the bacteria. Other strong cells like lymphocytes with round-shaped nucleus are of two types: B cells that shoot the enemy by antibodies and killer T cells that explode our infected cells. Look at the figures, cell 1, 2, and 3, and 4. Cell 1, the name of this leukocyte is macrophage. Why? Since it has a horseshoe-shaped nucleus as mentioned in the text. If the cell has a horseshoe-shaped nucleus, then it's a macrophage. If it has a multi-lobed nucleus, it's a granulocyte like cell 2. If it has a round nucleus, it's a lymphocyte like cell 3. Cell 3 and 4. Cell 3 is B lymphocyte and cell 4 is T lymphocyte. The mechanism used by cells 1 and 2 to kill the bacteria is called phagocytosis. They eat the bacterial cell and break it down. The job of cell 3, which is B lymphocyte, is to secrete antibodies, and the T lymphocyte explode infected cells. This is everything about the summary for week 2. Thank you for listening. Bye bye.